Every cup of coffee represents a dense, dynamic chemical matrix. This brew contains thousands of bioactive compounds, largely categorized into two families: alkaloids, including caffeine, and a diverse group of antioxidants known as polyphenols. Large-scale studies consistently show a strong link between regular coffee consumption and a lower risk of developing Alzheimer's disease. Because coffee contains such a high volume of simultaneous chemical interactions, scientists have struggled to determine which specific compounds are responsible for this protection. Researchers recently addressed this by using decaffeinated coffee as a biological control variable, allowing them to isolate the effects of caffeine from the rest of the matrix. This work relies on 43 years of longitudinal data from the Nurses' Health Study and Health Professionals Follow-up Study, analyzed at Harvard Medical School. Comparing clinical outcomes between caffeinated and decaffeinated drinkers allows us to map two entirely separate physiological pathways. The first pathway involves caffeine providing direct, targeted protection within the central nervous system. The second pathway relies on polyphenols, which modulate the gut-brain axis and the inner lining of the blood vessels. This model describes coffee as a dual-vector pharmacological intervention, with each family of compounds targeting different systems in the body. The evidence for caffeine-driven protection comes from a cohort study of 131,821 participants, followed for over 4 decades. The data shows that higher caffeinated coffee intake is significantly associated with a lower risk of incident dementia. This graph plots the risk of dementia against daily coffee intake. In the highest quartile of consumers, we see an 18% reduction in risk compared to those with the lowest intake. The association follows a nonlinear dose-response curve. The protective effect does not increase indefinitely with higher consumption. Instead, the neuroprotective benefit peaks at an intake of exactly 2 to 3 cups per day. This threshold corresponds to a daily caffeine dose of approximately 300 milligrams. This effect remained consistent even when researchers adjusted for genetic factors, such as the ApoE4 allele. They also factored in the polygenic risk score for Alzheimer's, which typically dictates an individual's baseline susceptibility to the disease. Even in those with high genetic susceptibility, the protection provided by a 300 milligram dose of caffeine remained significant. While caffeinated coffee shows a clear protective effect, verifying caffeine as the active agent requires comparing these results to the decaffeinated control group. In the same study, researchers analyzed participants who exclusively consumed decaffeinated coffee. While the caffeinated group shows a clear drop in risk, the decaffeinated data appears as a flat line. There is no association between decaf intake and lower dementia risk. This divergence isolates caffeine as the specific alkaloid responsible for protecting the central nervous system. To understand this result, we must look at how caffeine interacts with the brain's architecture. Caffeine's primary mechanism is the direct antagonism of adenosine A1 and A2A receptors. By occupying these receptors, caffeine blocks adenosine from binding to them. This blockade modulates synaptic transmission, helping to prevent the structural degradation associated with cognitive decline. Caffeine also influences biochemical pathways by suppressing beta and gamma secretase activity, which reduces the accumulation of toxic amyloid beta proteins. Without caffeine, the coffee matrix provides no structural protection against dementia pathology, leaving decaffeinated coffee neurologically inert in the brain. If caffeine handles neuroprotection, we must account for the high concentration of polyphenols that remain in both caffeinated and decaffeinated brews. These complex molecules are largely resistant to early digestion. They bypass the stomach and upper gastrointestinal tract to reach the lower intestine intact. In the lower intestine, polyphenols act as prebiotics. Prebiotics function as a selective food source for specific clusters of bacteria residing in the gut. When daily coffee intake is introduced, the architecture of the gut microbiome shifts. The population distributions change to reflect the new food source. A bacterial genus called Egertella expands aggressively when fed coffee polyphenols, utilizing them to synthesize specialized chemicals. These microbes digest the polyphenols and produce unique chemical messengers that the human body cannot manufacture independently. For example, these bacteria create indole-3 propionic acid, or IPA. This powerful antioxidant enters the bloodstream and circulates throughout the entire body. By outsourcing its biological impact to trillions of microbes, coffee turns the gut into an internal chemical factory. These new metabolites exit the gut and interface with the gut-brain axis, communicating with the brain through the vagus nerve. This microbial activity correlates with psychological data, showing that both caffeinated and decaffeinated coffee drinkers report lower levels of stress and depression. These same compounds interact with the endothelium, the inner lining of the blood vessels. Recent research, such as the Cosmos trial, highlights the difficulty of achieving these systemic benefits through isolated supplements. In the Cosmos study, isolated cocoa polyphenols failed to significantly reduce cardiovascular disease events in healthy subjects. Regular consumption of the whole coffee matrix, however, shows a strong link to a lower risk of heart failure and liver fibrosis. The decaffeinated control group represents a highly active, targeted therapeutic for the microbiome and the vascular system. We can now model coffee consumption as a dual vector intervention. One pathway carries caffeine to adenosine receptors in the brain, while the other uses polyphenols to trigger systemic metabolites through the gut. These two pathways operate in parallel, independently targeting neurodegeneration and systemic inflammation. This intricate system reaches a physical limit at the 2 to 3 cup threshold observed in the clinical data. This ceiling is determined by the kinetics of caffeine metabolism, which is governed by the liver enzyme CYP1A2. Enzymatic activity saturates at approximately 300 milligrams. Beyond this point, excess caffeine can increase anxiety and disrupt sleep, counteracting its neuroprotective benefits. The gut also has a limited capacity for processing polyphenols. Once the relevant microbial populations reach their conversion ceiling, extra intake provides no additional metabolic value. Consuming more than 3 cups per day provides no further benefit and can become counterproductive. Moderate daily consumption of caffeinated coffee remains the most effective protocol to support both brain structure and the health of the microbiome.